Hello, I'm going to talk to you today about the neuromuscular junction and muscle contraction. Before you watch this video, you should have watched a video that describes all of the pieces and parts of muscles so that you understand the things that make up the muscles, the structures within that muscle, and how those structures work together. Now what I'm going to do today is to tie all of those structures together and show you how those structures lead to a muscle contraction. So we're going to begin by talking about the neuromuscular junction. The neuromuscular junction is that place where the neuron meets the muscle. So if you look at this picture, and I think it's a very beautiful picture, you can see you've got muscle fibers. Okay, so each one of these pink-red structures is a muscle fiber. And then the brown structure coming here is the axon of a motor neuron coming out to form a neuromuscular junction with that muscle cell. All right, so now that we've got the basics, let's talk a little bit more about the things that are a part of that neuromuscular junction. So this is the site where an axon of a neuron, which is a presynaptic cell, meets a muscle fiber, which is a postsynaptic cell, and communication occurs between those two cells. Now what I mean by synapse is a space. So you see presynaptic cell, meaning before the synapse, and postsynaptic, meaning after the synapse. The synapse is actually a space. So for instance, if my hand is the muscle cell and my fist is the axon terminal of the motor neuron, there's a little tiny space between the two at the neuromuscular junction, and that space between the two is the synapse. All right, so here we have the axon of a motor neuron. Down here we have the muscle cell. Okay, the synaptic cleft is the same thing as the synapse. It's that space between the two. But within that end plate, that motor end plate, or the axon terminal, whichever term you use, of the motor neuron, you can see there are tiny little balls in there. Those balls are vesicles that contain neurotransmitters. A neurotransmitter is a chemical messenger that is made in a neuron, stored in a neuron, and secreted from that neuron where it causes something to happen on the next cell, which would be the postsynaptic cell. So within the vesicles we have here at the axon terminal of this motor neuron is the neurotransmitter acetylcholine. All right. Acetylcholine, as I mentioned, is that neurotransmitter that is stored in the vesicles. It's released when that neuron depolarizes. Now you should have already covered the action potential, so you're familiar with depolarization, and when the calcium rushes in at the axon terminal, it causes the synaptic vesicles to dump their contents. Well, now we're going to talk about those contents and what actually occurs. So acetylcholine is that neurotransmitter that's dumped into the synapse. Once it's dumped into the synapse, it's going to diffuse across and bind to these acetylcholine receptors. So down here we have the muscle cell, okay? Each one of these little structures here are receptors for that acetylcholine. When that acetylcholine binds, if you look down here, you can see that the channel becomes open. Okay, so this is a sodium channel. The acetylcholine binds the receptor. Okay, so acetylcholine is bound here. It causes this channel to open so that sodium can come into the muscle cell and potassium can be pumped out. And remember, when sodium comes into a cell, it's bringing that positive charge with it, and that is going to cause depolarization of that muscle cell, just as you saw depolarization occur with that motor neuron. So you're going to have depolarization going out in a wave across the membrane. Okay, the membrane is called the sarcoplasm or the sarcolemma on a muscle. So if your acetylcholine receptor is here, your sodium rushes in here, then you have depolarization going out, sort of like dominoes falling in a sequence. So going out and depolarizing that membrane. Now let's take that a little bit further. All right, so this picture should take you back to muscle structure. Okay, here is the membrane of that muscle cell. You're going to have your acetylcholine receptors that are a part of that membrane on the outer surface. Okay, your acetylcholine binds and you start depolarizing. Let me change colors here and I'll go with black. So we're going to start depolarizing all out through here, okay? From the receptor, where the receptor is, out in that wave. But you can also see that a part of that membrane invaginates down around the muscle fibrils similar to a bracelet. So like my watch encircles my arm, those extensions of the muscle membrane goes down and encircles the myofibrils. But because that is membrane, when that wave of depolarization reaches that, it's going to depolarize as well. Okay, that structure that I'm referring to as like my watch or my bracelet is called a T-tubule. Okay, so that T-tubule is where the membrane invaginates in and surrounds the muscle cell. All right, so as depolarization, that wave of depolarization travels along the membrane, when it gets to a T-tubule, it travels down that T-tubule as well. All right, so depolarizing, 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 right? Over and over and over as it travels along. Okay, now you'll also notice you've got this very web-like structure out here. Okay, this is your sarcoplasmic reticulum. And a part of that sarcoplasmic reticulum lies right beside that T-tubule and forms a triad. So you've got part of your sarcoplasmic reticulum here, part of your sarcoplasmic reticulum here, and there, of course, you have your T-tubule. So those three together, your T-tubule and your sarcoplasmic reticulum, are going to form that triad. Now, that sarcoplasmic reticulum is very important because it stores calcium. Okay, calcium is an ion, so it has a charge. Remember, it's going to cause things to happen. What happens when that calcium is dumped out? It's dumped out into all these spaces. Okay, so all these spaces are within the muscle cell, right? Now remember, within that muscle cell, you've got all of your myofibrils. You've got your actin and your myosin. Okay, so your actin and myosin are going to be within here. When that calcium is dumped into that muscle cell, it's going to bind to structures that play a role with that actin and myosin. Okay, so remember, the depolarization down and through the T-tubules causes the sarcoplasmic reticulum to dump calcium out into the cytoplasm of the cell. Within that cytoplasm of the cell, you have your myofibrils. Okay, now let's talk about what's going on. Remember, we've got the actin and the myosin. We've had calcium dumped out. So this structure, with its little golf club heads, is our myosin. Up here, the two-stranded pearl necklace is your actin. You have your ribbon-like tropomyosin, which covers the binding sites on the actin. Okay, now what? The binding sites are important because that myosin head that's lying back like this wants to attach to those binding sites, but it can't do so as long as that tropomyosin is covering the binding sites. And then you have proteins, a little 3-protein complex known as troponin. Troponin holds tropomyosin in place so that binding site stays covered. But when calcium comes in, calcium binds to the troponin. And as it binds to that troponin, it causes the troponin to change shapes. When it changes shapes, it knocks that ribbon of tropomyosin off the binding site. Once that binding site is open, then there is attraction for that myosin head to come up there and attach to it and form a structure known as a cross bridge. All right. A lot of stuff so far. Hang in there. All right. Calcium is dumped from the sarcoplasmic reticulum into the sarcoplasm. It binds to troponin, causes the troponin to change shapes. That knocks the tropomyosin off the binding sites. Now that binding site is open, okay? The binding site is open. That myosin head can attach to that binding site on the actin. Now you should also notice you've got ADP plus P bound to that myosin head. ADP, you should know what that is. Adenosine diphosphate, right? So ADP plus that inorganic phosphate makes ATP. Or you can take ATP and chop that phosphate off and make ADP plus phosphate, which is exactly what has happened here. We've got adenosine diphosphate plus a phosphate bound to that head. Now, once that myosin head binds to that actin binding site, that ADP and phosphate will drop off. when it drops off, it causes that head to bind tighter, and as it does so, it pulls against the actin filament. Now, in this picture, you see you've got myosin heads here, but you also are going to have the same thing going over here, where you're going to have myosin heads bound in this orientation as well to your actin. Okay, so each side of that sarcomere. Okay, the sarcomere, remember, is that little discrete structure within the muscle, the contractile unit. So you've got myosin heads here, and each of those sets of myosin heads are going to be bound to actin. When that ADP falls off, they pull because they bind tighter. And pulling on that actin filament shortens the sarcomere, okay, and leads to muscle contraction. All right, so far so good? Woo, it's a lot of stuff. Okay, now, see if I can get my words up here. Just bear with me a moment. Doesn't seem to be wanting to bring my stuff up. All right, so once that ADP falls off and you have that pulling, okay, that's where you have your filament sliding. All right, remember your ADP and phosphate fell off. Now ATP is going to come back in and bind to that myosin head again. All right, so you've got your myosin heads up here bound tightly where they've pulled against that and your muscle is contracted. ATP comes in and binds and breaks that cross bridge. So it's like the myosin head becomes too heavy, sort of. That's not really what happens, but that's a pretty good way to think about it. That myosin head gets knocked off of the actin when that ATP binds. So it pushes it back down into the resting position, ready to fire again. So you see here, your ATP binds. It's quickly going to be hydrolyzed or cut into ADP and phosphate again, and that's the way it holds on to that myosin head until the next wave of contraction occurs. All right, let's move on. All right, now let's talk about all of this together. Put it into the sarcomere shortening. Okay, here you have your myosins. Remember, you got heads going this way and heads going this way. Here's your actin filaments. Okay, so you've got all these myosin heads that are going to bind to those actin filaments once those binding sites are open. When they do so, remember, the ADP and phosphate drops off and the myosin heads pull. Okay, so when they start pulling, you can see you've got a shortening of that sarcomere. So when the muscle is fully contracted, you can see that sarcomere, your Z lines of your sarcomere, are now much closer together, so that muscle has contracted as those filaments slide against each other. Okay? Now once that ATP binds again, remember your myosin heads fall off and your sarcomere will relax again and lengthen. All right, so we've contracted that muscle. Now we've got to relax it. We can't keep that muscle in a constant state of contraction. What you've seen already is the role of ATP in muscle relaxation, because the ATP, remember, breaks that cross bridge that was formed between your actin and myosin. Okay, so that's one role of ATP. But now remember, we dumped all that calcium from the sarcoplasmic reticulum into the cytoplasm of that muscle cell. And as long as that calcium is present in that cytoplasm, then that troponin is going to bind to it, and it's going to keep that binding site open by pushing that tropomyosin out of the way. So as long as that binding site is open, that myosin head is going to keep attaching, keep forming cross bridges, and keep contracting that muscle. So in order to stop that, we've got to get that ATP out of the sarcoplasm. Get that calcium out of the sarcoplasm. It requires ATP to do that. So we have to have ATP to power a calcium pump that is going to pump that calcium back into the sarcoplasmic reticulum so that it can no longer bind to that troponin. The troponin goes back into its normal shape. The tropomyosin ribbon covers the binding sites, so there's nowhere for those myosin heads to attach. So that's two roles of ATP. One, it breaks the cross bridge, and two, it powers the calcium pump. But now remember, all of this started because we dumped acetylcholine into the synapse and it bound to the muscle cell. Okay, so we've got to get rid of that acetylcholine, or that muscle cell is going to continuously depolarize And as it continuously depolarizes, it's going to lead to that calcium being dumped and ultimately to muscle contraction. So we're going to use an enzyme called acetylcholine esterase. How do you know that's an enzyme? That's right, because it ends with ASE. Okay, so acetylcholine esterase breaks the acetylcholine into acetate, also known as acetic acid, and choline. The acetate diffuses throughout the synapse and is reused or broken down. The choline is taken back up into the motor neuron in a process known as reuptake. You're going to hear more about this when we talk about neurotransmitters. Reuptake, to take something back up into. So we're going to take that choline back up into the cell, combine it with an acetyl-CoA, and make acetylcholine, which then gets repackaged into a vesicle so that it can be dumped back out into the synapse for the next muscle contraction. All right, so a lot of material covered in a very short period of time. I do encourage you to watch this video more than once, to think about the roles of all of the players in this, and also to do the little animations and activities that we have to accompany this to help you understand it. If you have any questions, please be sure and ask me or your instructor. We'll be happy to talk to you about this and help you to understand what can be a complicated process. But what's amazing is to think about every time you move a muscle, this is going on. Have a good day. I'll talk to you soon.